Hello, everyone. This is Yamina from Dr. GPCR, and today I have with me Dr. Chris Dockendorf. We're going to be talking about his role at Function Therapeutics, but I wanted to mention before we get into the conversation that this is in collaboration with the 5th GPCR Targeted Drug Discovery Summit happening in Boston next week, and I'm excited to see Chris, the GPCR community, and everybody else coming together next week. Chris, welcome to it's not a podcast. Welcome to the interview.
Thank you. I appreciate it.
Very excited to have you on. Maybe we can start at the beginning. Can you please tell us a little bit about yourself and what you're currently working on at Function Therapeutics, obviously, if you can?
I'm the founder, CEO, CSO, whatever labels you want to do it. I'm Function Therapeutics. We're a very small firm here in Milwaukee, Wisconsin. And I wear a lot of different hats, but I guess probably the hat that I probably am most qualified to wear would be as a medicinal chemist. And so I've been hanging out in between the spaces between academia and industry for quite some time and kind of bouncing back and forth a little bit. But back in 2022, we actually opened the doors here. We're on campus at the University of Wisconsin, Milwaukee, which is a place with a beautiful new chemistry building. I was just telling you, we're watching the demolition of the old building, which a lot of people are interesting in listening to that. But yeah, I have a long interest in building small molecules that are going to be valuable for biology. I got into that when I was quite young, so I did an internship at the University of Waterloo. Did a series of internships when I was an undergraduate, and just kind of fell into doing organic chemistry and having biologists really excited about these small molecules we're making, and I just kind of taken it from there. And and I've been more and more connected with the GPCR community over the years for a variety of reasons, but really excited to be here.
Very excited to have you on, and you and I have been interacting indirectly on LinkedIn in the ecosystem, so I'm excited to meet you in person next week, but also learn a little bit more about your background and where you come from. So as we mentioned, you're coming to the 5th GPCR Targeted Drug Discovery Summit. What draw you to this meeting specifically?
Something I had seen last year. I think I saw the lineup and I was interested in it. And of course, I tend to be a lot busier than I like to be, and I get tied up with a lot of business-related stuff, and I can't attend as many conferences as I'd like. But that was one that definitely caught my interest. I think mostly from the combination of solid new industry, new companies focused on GPCRs along with academic groups. So that caught my eye. I actually made a note of it, hoping I could attend this year. And then actually Hanson Wade reached out to me and asked if I was interested in presenting. And I sort of said, I'd love to present, but we had lots of exciting stuff we can't yet talk about. So I kind of negotiated with them the opportunity to be at the page to lead a roundtable discussion, actually, which I'm really excited about because I expect to learn a lot from some people that are doing this 24/7, so to speak. So I'll be leading a roundtable focused on some interesting aspects to GPCR biology, including bias signaling, endosomal signaling, and GPCR oligomerization. So kind of 3 different topics that were just kind of hit some feedback. So
I think that's going to be very, very cool. And speaking of that, maybe I shouldn't plug it in here, but at the university next month, we're hosting Sudha Rajagopal, who's going to be talking about the importance of bias signaling, but also signaling away from the plasma membrane, including an endosome. So I'm curious to hear what the panel has to think about these topics. And it's a very exciting time to be working on GPCRs.
Absolutely.
The other thing I wanted to ask you, med chemists and GPCR pharmacologists most of the time don't speak the same language. What are some of the gaps between the two worlds that you're hoping to, help bridge at this meeting and beyond?
Really interesting question. I guess I might push back a little bit on the question to say that medicinal chemists, we tend to be jack of all trades in that we just want to get our hands dirty and build something useful, right? So if you can convince us that you have an interest, you have a valuable target, a valuable way to target it, most of us are pretty open-minded about how to do that. I think it's a super exciting time right now because, I was trained to be a card-carrying small molecule chemist, right? If you're building antibodies, well, you're not doing the building for you, so we don't want to do that. That's kind of out the window now. Chemists now, especially younger chemists, have been exposed to all these really exciting modalities. So you can have really small molecules, you can have larger molecules, things like we saw the recent approval of this GLP-1 agonist from Lilly, small molecule, argo, perhaps. I think I messed up. But then there's other modalities. There's ADCs. There's some peptides, antibodies. So there's a range of chemical spaces we play in this. And I think maybe one of the challenges is deciding, which chemical playground we play in. And obviously different chemists have different areas of expertise, but I'm seeing more and more situations where, even in my old lab, we were working on something where we have, one person building a peptide ligand for a particular receptor, another person building a small molecule that would be the dose separately or tie them together with a ligand. So yeah, I think from the medicinal chemistry standpoint, from that perspective, we just want to have a good starting point and justification for investing a lot of time and effort to build things. So the better the biologists are, I think, at maybe defining those needs, what a molecule needs to do, and where we start from, which is super important, because we can't necessarily just create something from scratch. We often need serendipity, or we're even seeing some computational leads emerge, obviously structural leads. Those are really important. So I'm seeing those conversations happening. I think that gap is getting smaller.
I'm glad to hear it. This question. Actually, I asked Dr. Terry Kinakin as well from his perspective, and I agree with him in the sense that for pharmacologists or for biologists, the chemists are our clients in a sense, and we need to provide you chemists the correct information and give you the hard numbers, basically. So I think a couple of months ago, we held an Ask Me Anything session within Terry's Corner, and his advice to biologists was, don't say anything to the chemists until you're sure about your data, because you do not want to have the conversation of the EC50 was this last week and it's that this week. So it's really about being diligent and making sure that your data is solid before going to the chemist, because you like numbers, you like precision, and those are, a molecule's property is a molecule's property. And when you work on GPCRs and cell-based systems, sometimes the stars align, sometimes they don't. But thank you. I'm glad to hear that from your perspective, the gap between medchem and biology or GPCR pharmacology is getting smaller.
So we're super excited to see the range of new tools that are available. I mean, even as a small company, we work with a number of different CROs, some of them which will be featured at the conference. And so we can get data that we couldn't have dreamed of. And certainly 10 years ago was not available. And if it was available, you really had to have a collaboration with one of the big academic that have commitments. And even in those cases, it can take a long time to get data. So things are a lot more, things are a lot better today. So those fast cycles, right, where you can get some data, you can design a molecule, synthesize it, you can submit it, you can get that assay data. So that data is important. But as Terry said, with that being said, yeah, sometimes that perfect data can lead you to go on a wild goose chase, because as chemists, sometimes we get overly excited about it. We compound, and we may not see all of its liabilities. Sometimes we get but it's hard to focus.
I agree. So in addition to the roundtable, that discussion that you'll be leading, is there any talk or a session or a question you're hoping this meeting helps you think through or learn about?
Well, great talks from newer companies, some platform-focused GPCR companies. I'm really excited to hear what they're up to. And also from some young academic labs that I'm not familiar with. I'm happy to learn more and maybe even have some future collaborations. I think for me, one of the big questions to ask is, what do we really need in terms of either structural information or some really good functional information to have a reliable chemical starting point for a project? We at Function Therapeutics, the work that we've been doing target primarily protease-activated receptors. Our leads started from serendipity, right? So actually puts found compounds that had really, really interesting functional effects. And I'm still seeing a need for that. I'm not seeing, I know people are advertising the ability to achieve to some binding sites, some compounds. I don't think we're there yet, but I'm certainly open to seeing exactly what is it we need as a starting point. Do you need a bit of structural data, and then we combine that with some computational work, identify the most promising binding sites and the most promising compounds. I just like to see what people are able to discuss in terms of their starting point. That would be. I'm enthusiastic about that, optimistic.
Absolutely. And I think what I like particularly about these meetings is that there's a nice balance between networking time and also talks. So it allows you to not only sit all day and taking notes from, somebody at the podium talking, but there's an opportunity to, while having lunch or having a coffee break or I think they have had in the past, actually, I know that in the past they've had explicit networking times. I'm assuming it's going to happen again this year. And I think it's a great opportunity to be able to go to someone and have these direct conversations, which to your point might lead to collaborations as well.
This will be my first meeting, so I'm the speaker list and in the company.
So outside of your work, going back to the science itself, what's a development in the field that, has your attention right now?
Super exciting. And as you know, I don't have to convince you about how exciting a time it is, but the fact that we have these amazing structural tools, really led by cryo-EM, computational tools are becoming more and more reliable. And then the functional readouts, the assays are becoming more and more available, right? So the combination of those three, it's just like down our biology and pushing drug discovery. So along those lines, I'd say, I don't know, if there's a lot of things I want to learn more about. I don't have the time as much time as I'd like. Having wearing an executive hat and dealing with accounting issues, all that part. So I don't have as much time to spend digging into some of the latest stuff. But one thing that I think caught my eye recently, and it's not a new problem, right? But the question is, how does the local membrane environment affect GPCR structure and function? And we've had some nice details about how function can be affected by membrane local structure in terms of, you have cholesterol nearby, you have lipid rafts, you have other protein co-receptors that can affect GPCRs. I think that's super interesting and really under addressed. But I guess I'm starting to see the possibilities of getting some structural information, some cryo-EM work that could point more definitively to how GPCR structure and function changes. That's something I'm keeping an eye on. I'm not sure if we'll dig into that too much for this particular meeting, but I think it's something that the field will be addressing, I think, increasingly.
I think it's important as well to develop those tools. There are some techniques and methodologies focused on developing these tools in order to better understand how does the native membrane environment influence GPCR function. To your point, we don't know nearly enough. All right, last question. If medicinal chemistry hadn't found you, what would you be doing?
That's interesting. I think I was really fortunate. My dad had a degree in physics, and he worked for decades as a meteorologist. So he didn't really push me in any one direction, but he just had these cool physics textbooks and stuff. I was fascinated by the physics and the astronomy kind of thing. So I guess maybe something in engineering maybe would have been a possibility. But I think looking back, one thing that I regret is paying little attention to biology, right? So, when you're a young person, you want to be in a club, right? And if you're in a club with chemists that just build molecules and don't care that much about biology, that's kind of get tunnel vision a little bit. So that's one thing that I wish I had paid more attention to when I was younger, was the biological aspects and medicine in general, right? So one thing I am a little bit, I definitely appreciate is the troubleshooting aspect. Maybe a little more for the patient clinic. Say, hey, this drug is problematic for you. I think this will be ideal, and let's look at pharmacology, and this might solve your problem. I like the idea that you can troubleshoot on a really short time scale and help people out at a short time scale. So maybe that would be attractive to me to go back and make medicine. Because time scales we're dealing with decades, right? Within the medicine, it's kind of crazy when I tell people how long you've been working on some of projects. But I'm just very grateful. And that's I just we collaborate with occasional people like our sharing topics, some other chemists, some chemists. I would trade that for the other opportunities.
Wonderful. So a scientist at heart, no matter what domain you end up in. Love it. Thank you, Chris. I really thank you for your time today. I'm excited to meet you in person next week. Bring a jacket. At least this week has been quite cold. I was seeing jokes around New England weather, and it is truly the case. Last week it was summer, and this week is winter is back.
I'll bring every possible jacket I can, but coming from Milwaukee, it may actually be summer.
That's actually what I wanted to mention. Coming from Milwaukee, this is nothing. This is summer here.
Absolutely.
Thank you so much. Really appreciate you.
Pleasure talking.